Hello, this is Genevieve Halstead-Johnson, and I will be presenting on the varying animal sensitivity to thalidomide. So why are some animals more sensitive to thalidomide than others? Humans and frogs, for instance, have shown the resulting congenital malformations due to thalidomide exposure of the embryo. However, rats exposed in embryo to thalidomide show no increase in congenital malformations. What makes humans and frogs more susceptible to thalidomide effects than rats? In our project, we will be utilizing frog, fruit fly, and rat embryos as animal models. We will have a control group untreated by thalidomide, while our experimental group is treated with thalidomide in embryo. We will tag the thalidomide with green dye in order to watch its progress within the embryos. This will resolve how the rat metabolizes the thalidomide rather than suffering its effects. Some background research on thalidomide. By utilizing animal-based research, one may predict what can happen in humans. Originally, there was no animal-based research done on thalidomide to have warned us of the impending tragedy of its usage in pregnant women to treat morning sickness. Usage in pregnant women resulted in many forms of congenital malformations in the children. Some were stillborn, others survived with deformities. Thalidomide's embryo toxicity is noted by Ikawa et al. In their 2013 study of it on human-induced pluripotent cells. Brent in 1964 studied pregnant rats and the effects by several different routes of thalidomide exposure during pregnancy. No malformations were reported. Meanwhile, Fort et al. In 2000 produced a study on frogs and thalidomide exposures, which had the resulting malformations of gut miscoiling, craniofacial defects, kinking of the notochord, abnormal limb bud organization, microencephaly with and without hydrocephalus, hind limb reduction deficit, abnormal mouth development, visceral hemorrhage, pericardial edema, stunting, incomplete eye development in the lens, hypognathia, abdominal hemorrhage, and cardiac defects. Our variables in this experiment are separated into 3 different variables. The controlled variables will be the thalidomide concentration, the embryonic stage when exposed to thalidomide. The independent variable will be the genus tested, therefore the frog, fruit fly, and rat embryos. The dependent variables will be the congenital malformations noted, presence versus absence, for instance, or the types of malformation. My hypothesis is that rats metabolize embryotoxins such as thalidomide in a way that they do not result in the congenital malformations seen in other genera such as frogs, humans, and fruit flies. The materials I will need for this experiment are the thalidomide, of course, the green dye to tag that, frog, fruit fly, and rat embryos. We will need plenty of these as we will have both the control group and we will have the experimental group, which will have several different stages that we mark. The embryonic fixative agent and a microscope to view those embryos. The steps for my procedure are: step 1, to select embryos for control and experimental groups and prepare the thalidomide with the green dye tag. Step 2 is to inject the experimental group embryos with thalidomide. Step 3 is to apply embryonic fixative agent to embryos throughout development. You will have several treated embryos of each genus. This will ensure that you can select at several stages to fix that embryo in place so you can see the progress of the thalidomide within the embryo. Step 4 is to observe the movement of the green dyed thalidomide throughout the development process in each genus of the embryos. These are the works that I cited, and thank you very much.